Down here, you can see that multiple codons can code for the same amino acid. What is a codon? A codon is just a 3-unit set of nucleotides that code for an amino acid. These are found in RNA. So by identifying what organism our cellulase came from, by the 16S procedure that Virginia mentioned earlier, we can take the organism, see what codons it uses for what amino acids, and manually turn the amino acid sequence into a chain of these codons, which will be our gene. Okay, so after that last step, we'll have primers made. And we are going to do some PCR on the original organism's DNA. Shown here in gray rectangles are the primers. And we will continuously replicate large pieces of these, of the gene that we want, the cellulase producing gene. And these large pieces are going to be called probes. I'll talk about probes in about 2 slides. First, we need a library to probe. A library is just a collection of genes from that single organism. These here you can see on the plates are E. coli that have been previously transformed with random genes you can find in our in the original organism. You can think of it as a library. A library has books, and each of these books have chapters. So this is our library. Each of these colonies is a book with a different set of chapters. So what we want to do is take one of those probes I mentioned earlier and radioactively label them. Then we subject the probes to the library and see which colonies show this radioactive flag. And whichever one does has our cellulase gene in it. And we can just pick that colony up. By now we should have our E. coli making tons of this cellulase. And I will now hand it over to Alea, who will talk about what we will do after this. That would include how to make this enzyme work better, how to improve its efficacy, and how to get it to be produced more, stuff like that. Thank you very much.
I'll say that. I guess. Wait. Okay. Okay. So, hi everybody. My name is Alea, and leaving off to what Vernon said, I'll be explaining about future references and basically what is supposed to be done next once we have our gene. Okay. So once we have our gene, we can insert it into a plasmid, which is also called an expression vector. So an expression plasmid is a piece of DNA that carries an inducible promoter. So we insert into the plasmid, which has an antibiotic resistance. So we have primers come in and cut the sticky ends, and then this will be our unknown bacteria or with the enzyme, and it can come in and look like this. And then once we have that, we can insert into E. coli by doing a step called transformation. And once we insert it into E. coli, it can replicate, so we can make more, which is cloning. And then we can plate it onto plates and put sealing plates so that only our bacteria that have our plasmid are able to grow, which are the pink ones, and the white ones are obviously bacteria that do not have our plasmid, which will die. So then yeah, so it'll replicate, make more, then we can put it into, make liquid media and put into our culture and then do purification. So yeah, okay. So once you do that, okay, we can purify the enzyme. In order to purify the enzyme, we can do a procedure called affinity chromatography. Why? Because E. coli will produce this enzyme intracellular and not extracellular. We will need to separate the protein from all the other enzymes and proteins. Hopefully we can do that. Hopefully once we have purified this enzyme, we can determine the native activity. So this is just how affinity chromatography works. So these are all the enzymes. So yeah, we can get rid of all of the enzymes that we don't want, which is this step, and then keep our enzymes in the next step. Okay, so determining the native activity. So me, my team, and I studied a cellulose producer, which was Aspergillus niger. As you can see the graph, you see the activity compared to the mass of the enzyme. So yeah, this is the mass and the activity. So the graph also shows the study of Aspergillus niger that, okay, yeah. We will need to determine the native activity once we've purified our enzyme. So we just use Aspergillus niger as just a comparison to what we were going to do because our plates weren't ready. So yeah, this is something that we were going to do as well. So improving the activity. So however, once we determine the native activity, we are able to check for improvement within the activity, which is what directed evolution and what, hold on. Okay, yeah, so this is the meaning of directed evolution. So therefore, well, these are the steps. So once we can select a gene and then create variants, libraries of it, and then insert it into a plasmid, and then insert it into E. coli, and then screen it, look at it, and then just repeat the steps over and over again to show improvement in the activity. And once we do that, we can do gene shuffling. So this is a process of mixing our unknown gene with similar enzymes so that we can see if by doing this, is there a chance of increasing the activity. For example, it's like playing Uno, and you have, so each person is supposed to have 7 cards. So say you have 7 yellow cards and your opponent puts down a red card, and it's not a number that you don't have and it's not a color you don't have. So therefore they have no other choice but to draw or to get a new stack of cards. So this is what gene shuffling is like. So yeah. Okay. So what we hope our graph to look. So say we gene shuffle Aspergillus niger, we can do the same procedure. We can do the same procedure and plot on the graph to see if there's an improvement. So say this is Aspergillus niger and we do gene shuffling, and then this is what it may look like. Oh man. Okay, so E. coli and yeast. So people may say, why E. coli or yeast? Because E. coli and yeast are 2 main organisms that are used here at J-Bay, and we know everything about them, which makes it easier for us to maintain food. Also, okay. So also, okay. Wait. So E. coli and yeast are they can convert into biofuels. Well, but it would be E. coli and yeast can already convert into biofuels. Therefore, as Martine said, we are at this step, and we're trying to get we're trying to get the E. coli to break down the cellulose itself and then make it instead of going through these processes, we're trying to go straight to biofuels. And what else? So yeah, and if it goes from here to here, it will possibly be more cheaper and more easier. And yeah, so now all questions are allowed to be asked.